DNA polymerase is a critical enzyme for cellular function, but it can't initiate replication. We've talked about the fact that you have to have primase synthesize an RNA fragment to begin the process. In vitro, it's common to begin the process with single-strand DNA fragments. But you have to add at the 3' end of an existing DNA or RNA for DNA polymerase to work. You have single strands with a primer that are copied 5' to 3', and only 5' to 3'. It will use single-stranded DNA as a template. DNA polymerase will not copy an RNA template. It uses base pairing to identify partner nucleotides. And the incoming nucleotides have to have 3 phosphates and deoxyribose to be used by DNA polymerase. It forms a sugar phosphate linkage, so it links the 3' hydroxyl of one nucleotide through a phosphate to the 5' hydroxyl of the next nucleotide. This process is driven by the hydrolysis of the nucleoside triphosphate. Not just the release of pyrophosphate, but also the hydrolysis of that pyrophosphate to inorganic phosphates gives energy from the high energy bonds to drive this process. And it makes the process processive in the direction of synthesis. So the energy release keeps this moving forward. But it requires a primer, a 3' end. It cannot initiate on its own. Actually, there are lots of DNA polymerases in cells, including in bacteria. DNA polymerases have specialized roles. DNA polymerase III in bacteria is dedicated to replication, a highly processive leading strand DNA polymerase. Sometimes this enzyme will work in DNA repair. Some DNA polymerases only work in repair. Remember that DNA polymerase I is much better at removing the RNA segments that are part of Okazaki fragments. The DNA polymerases are often labeled using either the Greek alphabet or Roman numerals to distinguish them. There are 7 different families of DNA polymerases, and they have distinct structures. But there are features that are common to all DNA polymerases. The main ones to remember here are DNA polymerase III, which is highly processive, and DNA polymerase I, which is good at removing RNA and important for the lagging strand. One of the features that DNA polymerases have in common is proofreading. The capacity of DNA polymerase to proofread is very important. If you have a mismatch and DNA polymerase continues, you're stuck with the mismatch. But with the ability to proofread, it can detect and remove mismatches and replace the wrong base with the correct one. This ability is paramount for high fidelity, the faithful copying of DNA during a replication process. So if DNA polymerase gets stuck, it detects a mismatch, it cannot go forward. The proofreading capacity has to do with the shape of the active site of the protein. It fits around a base pair to confirm that it is correctly sized in the binding site. If not, a 3' to 5' exonuclease activity, which every DNA polymerase has, is activated. And this removes the mismatched base pair and allows a correctly matched pair. In this case, a correctly matched GC pair. And then it returns to extension mode for DNA polymerase. So all DNA polymerases have this proofreading function. They're able to recognize when the base that's introduced is in error. There are 3 activities associated with DNA polymerase. The first is polymerization, 5' to 3' for synthesis. Then there's 3' to 5' exonuclease that is able to remove an erroneous base and can then go forward with its 5' to 3' polymerization. And interestingly, most DNA polymerases have a 5' to 3' exonuclease to remove DNA and RNA in front of synthesis. This is used for removal of the RNA primers during replication, but often this activity extends into the DNA and it can help correct any mismatches in the error-prone initial steps of synthesis. In order to keep things going, there's a protein called a clamp. And you can see the structure here. It surrounds the double-stranded DNA and it moves behind the DNA polymerase to get the strands together. And to ensure that you have time to be correctly paired as this DNA polymerase moves down the strands. And it also ensures that long segments of DNA are synthesized before DNA polymerase on the leading strand comes off. So this is most often associated with DNA polymerase III and most often associated with the leading strand because the continuity of that synthesis is critical. It surrounds the DNA and creates a pocket in which the base pairs are more likely to form because they're surrounded. So the DNA clamp follows DNA polymerase III on the leading and lagging strands, ensuring that the double helix and the base pairs that stabilize it are formed correctly following the process of replication. I'd like you to think about two things. Why is DNA replication only 5' to 3'? Could you envision a mechanism by which it could go 3' to 5'?